Good morning students. Today we are going to see about ICH and WHO guidelines for validation and calibration of equipments. Validation and calibration. Validation means the action of proving and documenting that any process or procedure or method actually consistently leads to expected results. So validation means it is a process of documentation of some procedures or method or equipment that already exist and which if we done any calculations we will get the expected results. Calibration. It is the set of operations that establish under specified conditions the relationship between the values indicated by the instrument or system for measuring represented by material measure and the corresponding known values of reference standard. So calibration is the set of operations that established under the specified condition. It is used for measuring example weight, temperature, pH and recording the controlling of values and material measures corresponding to their known reference standard. WHO guidelines. Equipment validation and qualification includes design qualification, installation qualification, operational qualification, performance qualification. Design qualification. It is also called as DQ, which defines the functional and operating specification of instruments and details for the decision in selection of the suppliers. So it is design deals with the first step of functional and operating specifications. It includes description of analysis problem, description of intended use of equipments, description of intended environment, preliminary selection and functional performance specifications. So first what and the analysis problem that must be described with the design. Then the use of equipments, then description of where the preparations have been done, then environmental factors, and the preliminary selection function of the specifications like technical, safety, etc. Next one, installation qualification. Installation qualification established the instrument is received as designed in specific. It is properly installed in the selected environment and this environment is suitable for operation and use of the environment. So installation means first design, then we have to design the instrument and we should place in the particular area which is suitable to the environment and it is properly installed in the specific place and it should be used for the instrument. Supporting groups, plant engineering group, equipment management group. Operational qualification. Third one is operational qualification. It is the process of demonstrating an instrument with the functions according to the operational specification in the selected environment. So it is the third part of the validation which demonstrate operationally which demonstrated how the instrument works with respect to the surrounding of the environment. It includes application of standard operating procedures, utilization list, process description, test instrument utilized to conduct test instrument calibration, critical parameters, test function summaries. So these operational qualifications include application of standard operating procedures, utilization of list, process description, test instrument calibration, clinical parameters, test function and test function summaries. Performance qualification. Performance qualification is the process of demonstrating instrument performs according to the routine use. Performance means how the instruments get work according to the routine use. We must check the instruments capacity and their readings before we are doing any production or manufacturing. Next, ICH guidelines. ICH guidelines, International Council of Harmonization, defines typical analytical validation characteristics to do test to apply them and examples the how to. So it is used in typical analytical validation to test and apply the examples and how they work. It includes specificity and detection limit. Specificity. Specificity is the ability to access unequivocal the analytes present in the components which may be expected to be present. So specificity na ability to components having ability to access the analyte is called specificity. Detection limit. Detection means we are finding something and it has a specific exact limit value of an individual analytical procedure is lowest amount of the analyte in a sample which can be detected and not found with exact values. So detection limit means if any analytical procedure the lowest amount of analyte in a sample can be recorded. Third one, accuracy. Accuracy means closest value of the true value. Accuracy of an analytical procedure express the closest agreement between value and accept their conventional true value and reference value and the value form. This is sometimes termed trueness. So accuracy means it is the closest value which is accepted to the true value or reference value. Precision. It is the closest to the agreement between series of measurement obtained by multiple sampling of same homogeneous sample under the prescribed conditions. Prescription may be considered as three-level repeatability, intermediate precision, and reproducibility. So precision means the closest value nearer to the agreement. So degree of scattering the effect. So it may be of three levels: repeatability, how much time it is repeated, intermediate precision, and reproducing. Again it is reproducing. Fifth one is linearity. Linearity means it is the straight and direct value obtained in a calculation or result. Linearity of is an analytical procedure. It is the ability to obtain test results which is directly proportional to the concentration of analyte in a sample. So linearity means, as we said, it is the linear value of a given data, which means it is directly proportional to the concentration, which means the amount of analyte and which is present in a sample. Robustness. Robustness means it is the measure of its capacity to remain unaffected by small but deliberate variations in method parameters, provides an indication of reliability during normal usage. Robust means it is the measure of capacity some unaffected by small. It is the measure of capacity, some substance capacity to be unaffected by any external environments due to their normal usage. Example, change of pH in mobile phase. Second one, change of column. And the third one, liquid bit change temperature. pH change, change of the column and change of temperature. Range. Seventh one is range. Range is an analytical procedure which is the interval between upper and lower concentration of an analyte sample for which has been demonstrated that analytical procedures suitable level of precision, accuracy, and linearity. Range means the concentration from higher concentration to lower concentration of the sample, analyte present in a sample. This includes maximum the demonstrated procedure of analytical procedure suitable for level of precision, accuracy, and linearity. So this nicely the graph of range is given. It is a response amount curve with a linearity range. Eighth one, quantitation limit. Quantitation means amount of analyte. So it is an analytical procedure in which lowers the amount of analyte in a sample which can be quantitatively determined with suitable precision and accuracy. The quantitation limit is parameter of quantitative assay for low levels of compound in sample matrices and is used particularly for determination of impurities or degradation products. So, quantitation limit means it is the procedure in which lowest amount of analyte in the sample is determined. We have low amount of analyte determined with the wedge test means quantitation limit. Okay. It determines the parameters of quantitative assay for low level of the compounds of sample matrices. So this determines the limit of parameters for low level of compounds. Thank you.